My name is Brian Munoz. Yes, another Munoz here in the podium, but besides Dr. Miguel Munoz and I share the same last name, come from the same country. We are talking about bananas. No, we're not related. Spartanocarpic. My presentation, well, I'm a first-year PhD student. I belong to the Nutrigenomics lab under the direction of Dr. Massimo Iorizo. And today I'm going to be talking about the whole biodiversity that Dr. Munoz spoke about and how are we using and studying that with our team. So the first thing I want to talk about is this, a simple banana. Every time you look one of these, you never really think about what is all the work there is behind this. It's just a simple, small fruit. Everybody eats it. You have a bunch in your house. They go spoil pretty soon, and that's pretty much about it. If you're just like me, when you think about bananas, I think about this. Breakfast. That's the first thing I eat for breakfast. I like it much this way. Some other people may think about bananas this way, desserts. Why? Because it has a very distinct flavor. So you will have your banana cakes and banana breads and banana cookies and banana ice cream. All these bananas flavored because it's affordable, it's sweet, and you know what are you gonna get. And somehow, sometimes you think about bananas this way. Why? Because bananas have made it to the stream media through the pop culture. It's so part of our lives that we never really think about what is behind all this. But there are some other uses for banana that we know little of. For example, did you know that banana fibers have buoyancy and they are water repellent? So they are perfect for making ropes for fishermen and ships. Also, because of the same properties, they are perfect for making boxes for storing things you want to keep dry. The fibers have been also used for paper, and the banana paper now is being used for fruit packing, such as tea bags or paper bags, Ziploc bags. And one of the little-known uses of banana is for textiles. Yes, they do textile from banana fiber. Yes, there are clothes made out of banana fiber. Now, we may think these uses are pretty much new, novel, or different. But in fact, they are not. Back in the 15th century in Japan, during the Edo period, Japanese used to make kimonos out of the banana fibers because it was lightweight and it was breathable, so they were perfect for the summers. And also, Latin Americans have been wrapping our food in banana leaves for decades. So yeah, it's really just a lot of re-engineering, but these are all ideas renewed. So how did all started? And I'm just making a summary of everything that Dr. Munoz talked to us about it this morning. So it starts in this region of the world, Southeast Asia, with these two species, Musa cuminata, which you can see right here. I know it looks big on the screen, but it's actually, it's this little bitty thing. And then we have Musa balbuciana. Again, it's this little bitty thing. This is what we could actually call the mother and the father of all bananas in the world. Musa balbuciana was pretty much bred, domesticated 7,000 years ago, as well as acuminata, but they were bred in the islands of the South Pacific area, while Musa cuminata was bred, reproduced, and domesticated in the southern part of China. The hybrids and all the mixes made way to India, Africa, Australia, and from Africa came to America. But interestingly enough, one of the most important movements of banana that happened over the centuries was from Africa to Europe. And you may have to remember these two guys, Sir Joseph Paxton And of course, now you know why. William Cavendish, Duke of Devonshire. They got a gift from a friend from the island of Mauritius that is in the Indian Sea. And Sir Joseph Paxton was an architect, botanist, and gardener. Yes, a lot of things. And he used to work for William Cavendish. So he was the first one to describe this variety. And in honor of his protector, he decided to call this variety Cavendish. So it's because of them, they decided to spread this variety of banana all over different botanical gardens around the world. And they finally banana arrived to America, first at the Caribbean, and from the Caribbean, it moved to Central America, where it became a very important crop. Now, when we think about banana, and now we're going all the way back to the 1800s, bananas were not really what we know now. They were not the yellow, very nice type of banana that we eat now. It wasn't what it was around back then. Usually, the bananas they were eating, they were cooking bananas, and they were usually green or red. They were a specialty. They were a novelty. They used to be wrapped in tin foil. They used to be very, very expensive. People would eat it on a plate with a fork. And people needed instructions on how to eat these exotic, weird fruits. Because they were very expensive, and honestly, they weren't that all good. You know, people used to prefer the more varieties that they were used to eat, like peaches and apple. But we all know the history of the United States, and during the last years of 1800s to the beginnings of the 1900s, you know, Civil War happened, civil distress happened, the economic downfall happened. So apples and peaches prices started to go up, while banana prices started to go down. And that was key for the banana dispersion of the world, because bananas were so cheap because they were produced in the Caribbean and Central America, where there was neither of these problems, that people started preferring bananas over apples and peaches, making it the fourth largest fruit ever, or well, actually agricultural product ever consumed in the world. But bananas are beyond that, because for countries like us, Costa Rica, bananas made a big impact in our way of life. Bananas used to be very labor intensive. The bunches were very heavy. They were carried by men or by donkeys. But with the banana and the banana money came the railroads, and schools came, and town came, and a lot of economic growth came. Even that much that banana was depicted in one of our most precious bills. And this bill is something to remember, because the guy holding the banana bunch, it's backwards. So it's not the way it is supposed to be, and also bananas are not grown on the beach. But anyway. But as Dr. Munoz mentioned before, disaster came along in the name of Panama Disease. That was during the 50s and the 60s, and pretty much did all this to the banana plantations. But if you remember this guy with this banana that he dispersed, people started to noticing that this banana was the only one not dying from all that. So this is the banana that we eat now, pretty much by chance, not really by choice. So the story started here with Acuminata balbisiana. After years of cultivation, now we get something like this. It's even bigger than us. And what our research is trying to do is trying to tap over this biodiversity of banana and focus on two main things: nutritional profile and disease resistance, so it won't happen what happened before. And without further ado, I would like to introduce my wonderful team of interns. I have Viri Manduhanu, Andrew Ratchford, and Devon Schultes. And I'm going to leave the floor to them.
Okay. Thank you, Brian. So as we heard before, there is a lot of biodiversity in bananas, and cultivated in over 130 different countries, it is the second largest produced fruit in the world. 2 months ago, Brian was in Costa Rica collecting different varieties of bananas from those banana collections for us to analyze. And those banana collections consist of approximately 250 different varieties of banana. And not only do they differ in size, shape, and color, but they may also have a different content in phenolics, sugar, vitamin C content. Because of all this diversity, our study goal was to investigate different metabolites from both the pulp and the peel of banana, and also to compare metabolite profiles in natural and chamber maturation stages. These are the 5 different accessions that we decided to focus on. We had Cavendish, Baby Kai, KKB, PK Lin, and Prata. From left to right, we have ripening stages, which are stage A, B, and C for chamber maturation, and then we have another stage C for natural maturation. The 2 ripening methods, we have chamber maturation and natural maturation. For chamber maturation is the most commonly used. It's a chamber that's filled with ethylene, which quickens the ripening process of bananas. And for natural maturation, it's a longer process, and it would never be adapted by the industry because it faces many dangers like the fruit falling or animals attacking the fruit. This is the sampling strategy that is going to be used throughout this entire project. For our particular project this summer, we only focus on the 5 accessions that we mentioned before. We had 3 different clones. We had natural maturation and chamber maturation. For natural maturation, we use stage C. And for chamber maturation, we use stage A, B, and C. And we analyze both the pulp and the peel for these stages.
So the metabolites of focus this summer, like Viri said, were sugar, vitamin C, and total phenolics, each of these playing a very crucial and important role in a banana. So the methodology we went about by analyzing and quantifying these contents in the banana accessions that we studied this summer all had different methods. So with total phenolics, sugar, and vitamin C, you can see here we used the freeze-dried pulp and peel of each of the accessions, with sugar being the exception where we only tested the pulp. So with total phenolics, we used our Synergy H1 microplate reader, being a little bit different than sugar and vitamin C, where we used HPLC to analyze our data. So to analyze a little bit into the metabolites that we went into this summer, sugars being the first. Sugars obviously impact the taste of the bananas, so how good the banana tastes to the consumers, but it also has nutritional significance, affecting the caloric content of the bananas. We tested individually sugars as glucose, sucrose, and fructose. As we can see right here on the right, we have the results from our data collection. Natural maturation showed a significantly higher amount of sugar content in all of the 5 accessions that we studied this summer. We can attribute this to the longer time the fruit has to stay on the plant and absorb the sugars. This is also a really great example of how natural maturation can give us the full nutritional profile of the bananas that we studied. So moving on to total phenolics relates to the antioxidant activity that goes on from the banana in the human body. It has industry and healthcare benefits, and we can relate the phenol content to the health benefits of that accession. So looking at the results that we received from all of our data this summer, we can see that the peel has a much higher content of total phenols than the pulp. This shows that maybe the peel also can be just as useful as the pulp. So the peel is often thrown away when we consume a banana, but if we notice that there are more nutritional benefits to the peel, we can somehow utilize this and exploit the utilization to the consumer. Vitamin C is crucial for growth and repair of human tissue. So if the extract is removed from the banana peel and applied topically, it can be helped for healing just skin wounds. It also has protective properties against certain diseases like gastric cancer. And this was a metabolite that we studied and found significant results on as well. And we see that in almost all of the accessions, the natural maturation had a significantly lower amount of vitamin C in all of the accessions. And this is interesting because we can relate vitamin C's sensitivity to light to the exposure that, of sunlight it received on the plant by staying there to ripen and finishing the ripening process there. And now I'm going to hand it over to Andrew to talk about how these metabolites data were entered into our database.
Hi, thank you. As you can imagine from our previous slides, the sheer amount of data from the nutrient profiles is amazing. In order to sort of facilitate the production of this project, we have to organize this data into a central location that is easily accessible to all people. Here we see that BananaDB is our database of choice. We've created a 3-tier, multi-level system for data entry. So at any point in time, someone in the field in Costa Rica can introduce the phenotypic data about the fruit itself. Someone in the lab can talk about the nutrient profiles and introduce that, as well as someone somewhere else, anywhere in the world that has access, can do data curation and organize it, as well as make it visually appealing. We have 3 ways to sort of visualize this data. We use QR codes inside of the lab to determine which sample is inside of each capsule that we're studying. That helps us keep track of what nutrient profiles are available in each. We also have a data pool and a combinational query that lets us search and pull out specific things that we're searching for in our total sample list. Moving on with the database in the future, we'd like to sort of introduce some data about it that contributes to the resistance of black Sigatoka in banana. Cavendish banana specifically, are very heavily affected by black Sigatoka. In 1 year, up to 50% of a crop can be wiped out by this disease, so it's very important that we take a hard stance on this subject. The treatments now are mainly fungicidal, so coating the leaves in a special fungicide to prevent transmittance of the fungus. So that's very difficult because banana leaves shed water. Instead, we're taking a genomics approach with this project and looking for the upregulation and downregulation in genes that lead to Sigatoka resistance. Because of this, we're looking for DNA extraction methods that will help in our genome sequencing. The DNA extraction methods here will yield DNA that will be used to look for those specific genes that lead to the resistance or susceptibility of Sigatoka in the specific accessions that we ourselves have chosen. We have 3 methods here, the CTAB, the nuclei extraction, and the QIAGEN DNA kit. The DNA kit is a very tried and true method. It's very pure, very fast, very easy, so it was an easy choice. The nuclei extraction method, it extracts the whole nucleus, so it prevents DNA, sort of DNA interference from the other organelles, so that leads to the overall DNA extraction results. The nuclei extraction method actually extracted base pairs of up to 5 times the length of the DNA extraction kit, as well as the CTAB actually only produced RNA, whereas in carrot, blueberry, and melon, it produced DNA. So the nuclei extraction method will be the method we're using going forward and hoping to sequence genomes with in the future. As for now, I'll hand it over to Brian as he concludes our presentation.
Again, I just want to say thank you to my wonderful team. They've been great support for me during the summer, and they helped me to advance my research a lot. As well, there is a lot of people I would like to thank. It's impossible for me to put them all in this place and to put them, to name them all right now. But I have a wonderful team of people backing me up at the Adult Nutrition Lab, at the Adult Division, Banana Division in Costa Rica, at our lab that has been supporting different stages of this project. If you have any questions, don't forget to look for us. We're going to be at poster 1 and 2. We'll be showcasing some more banana, and please come visit us. Have a wonderful morning. Bye.